Welcome to Lecture 8. Lecture 8 is on the physiological and ecological considerations of photosynthesis. Please refer to the recommended reading from Tays and Zeiger Chapter 9 for complementary information. This subject is offered at NMIT as part of the agricultural and the viticultural degrees. My name is Nikki Cooley. In part 2 of this lecture, we are going to look at the impacts of temperature on photosynthesis and also the impacts of carbon dioxide and carbon dioxide concentrations and photosynthesis. So let us begin with the responses to temperature. The heat load on a leaf would be enormous if all the solar energy was absorbed and no heat was lost. The heat load is dissipated by the emission of long-wave radiation, approximately 10,000 nanometers, by sensible heat loss and by evaporative or latent heat loss strategies. In the image below, we can see a visual representation of energy input and the mechanisms of heat dispersal that are available to the plants. Exploring these three strategies in more detail, starting with radiative heat loss. This is where all the objectives emit radiation in proportion to their temperature. Sensible heat loss is where the temperature of the leaf is higher than that of the air. The heat is converted or transferred from the leaf to the air. And finally, latent heat loss works on the similar mechanisms to evaporative cooling. The evaporation of the water requires energy. So when energy evaporates from a leaf or during transpiration, it withdraws large amounts of heat from the leaf and cools it. The relationship of photosynthesis as a function of temperature is described as a bell shape, and this is shown for C3 and C4 plants in the figure below. However, two contrasting responses reflected by temperature, optimum when each species grown in its own habitat. For example, the example of the hot desert C4 is more optimal at a higher temperature, just over 40 degrees, than the example of the cool coastal C3 plant. CO2 diffuses, described here as CO2d, from the atmosphere into the leaves through the stomata into the intercellular air spaces, then into the cells and finally into the chloroplast. Just as high CO2 diffusion is photosynthetic limiting, so is low CO2 diffusion. CO2 diffusion is a trace gas with an approximate concentration of 0.039% of air. Because diffusion rates depend on the concentration gradients in leaves, appropriate gradients are needed to ensure adequate diffusion from leaf surface to the chloroplast. The cuticle is nearly impermeable to carbon dioxide diffusion, so the main entry is via the stomatal pore. CO2 diffusion into the air spaces between the mesophyll cells is in the gaseous phase, while the remainder of the diffusion pathway into the chloroplast is in the liquid phase. Sharing the stomatal entry pathway by carbon dioxide and water presents a functional dilemma. In high humidity, the carbon dioxide diffusion gradient that drives water loss is about 50 times larger than the gradient driving carbon dioxide uptake. In dry air, the difference is greater. Thus, a decrease in stomatal resistance through stomatal opening facilitates higher carbon dioxide uptake, but higher water loss. There are three main points of resistance of the supply of CO2. The first is the boundary layer, the second the stomata, and the third the intracellular spaces in the leaf. The boundary layer results in unstirred air around the leaf surface. This is known as the boundary layer resistance, and it decreases with leaf size. Stomatal resistance is the main limitation to carbon dioxide diffusion, while the resistance incurred in the intracellular spaces is small. So where inside a leaf do the maximum rates of photosynthesis occur? The light enters the upper surface of the leaf. Blue and red photons are preferably absorbed by chloroplasts near the surface, while green photons, or light, penetrates deeper into the leaf and is effective in supplying energy within the leaf, depleted of the blue and the red light. The figure on the screen describes the efficiency of light absorption at different wavelengths of light. This is the evidence for the statements made in the previous slide. Different wavelengths are absorbed at different depths in the plant tissue. For many crops grown in controlled environments such as glasshouses or greenhouses, optimal water, nutrition and enriched CO2 can result in increased productivity. Photosynthesis can be expressed as the partial pressure of carbon dioxide in the leaf, and this is referred to as CI. This enables evaluation of photosynthetic limitations. Increasing carbon dioxide concentration at which photosynthesis and respiration balance each other defines the carbon dioxide compensation point. That is, the point at which there is no net flux of carbon dioxide from the leaf. When you look at the figure on the screen and examine it in more detail, it can reveal the following. In C4 plants, photosynthetic rates saturate at internal carbon dioxide values of 15, reflecting the effective carbon dioxide concentration mechanisms. In C3 plants, increasing CI levels stimulate photosynthesis over a broader carbon dioxide range. In C4 plants, the CO2 compensation points is nearly zero, reflecting low levels of photorespiration, while in C3 plants, the carbon dioxide compensation point is about 10, reflecting carbon dioxide production due to photorespiration. The screen on the slide reflects atmospheric carbon dioxide concentrations at previous stages throughout history. The figure on the far left shows that many years ago carbon dioxide levels fluctuate quite a lot. From 1960 there has been a gradual increase of carbon dioxide levels. Responses from the plot of carbon dioxide compensation points indicate that C3 plants may benefit from increasing atmospheric carbon dioxide concentrations. Photosynthesis in C4 plants is carbon dioxide saturated at low concentrations. Therefore, there will be little benefit to this group of plants with increased carbon dioxide concentrations. The ancestral photosynthetic pathway is the C3 photosynthetic pathway. During geological time periods, when the carbon dioxide concentrations were higher, carbon dioxide diffusion through stomata would have resulted in higher CI values, thus higher photosynthetic rates. Although photosynthesis in C3 plants is carbon dioxide limited, they account for approximately 70% of the world's primary productivity. While C4 photosynthesis is an evolutionary adaptation and evolved more recently, between 10 and 15 million years ago. In recent years, there have been much debate about carbon dioxide concentrations in the atmosphere and how they may increase into the future years. So, assuming they do increase, how will photosynthesis and respiration be affected by the increases of carbon dioxide concentration levels at 400 parts per million or more? In order to try and answer this, scientists have been doing experiments on free air carbon dioxide enrichment, or called FACE for short. These experiments involve chambers or areas where plants are subjected to both normal or current day carbon dioxide levels and other containers or areas where the carbon dioxide level is increased. This can provide new insights into how plants will respond. For example, 20% increases in photosynthetic rates in C3 plants and none in C4 plants. Plant can grow quickly and growth may be reduced by nutrient availability. The figure on the slide is a visual representation of one of these FACE or free air carbon dioxide enrichment experiments. Here you can see chambers or areas where carbon dioxide is delivered to the plants, ensuring higher levels. The growth and the development of the plants are then compared. This FACE experiment in soybeans field in Illinois shows that there are areas of where the enriched CO2 is delivered, results in warmer conditions, an average warmer temperature of 27.5 degrees compared to the ambient CO2 where plant temperature is 26.1 degrees. If you have struggled with any of the concepts of photosynthesis in this lecture, then I have 2 alternative YouTube videos on photosynthesis that you can watch, so please refer to the link on the screen. In summary, I hope that you now understand about the heat loss strategies that are available to plants to optimize photosynthesis. We've touched on the impacts of temperature and examined how they are dependent on the carbon fixation mechanism. We've looked at the entry of carbon dioxide into the plant and the role of the transpirational compromise and stomata. We've examined and learnt about internal carbon dioxide levels, CI, and how they reflect photosynthetic efficiencies. And we've also touched on the future climates and how photosynthesis may be impacted. This brings us to the end of this lecture and to the end of the lecture series on photosynthesis.